Hello, everyone. My name is Mark Milhaven. I'm a fifth-year PhD student, or incoming fifth-year PhD student at Arizona State University in Suzanne Pfeiffer's lab. And today I want to talk about a tool that I've been working on for a little while that helps recommend de novo mutation threshold filters when estimating mutation rates. So the first thing I want to talk about is the importance of mutation rates. The Pfeiffer lab primarily studies mutation rate variation in non-human primates. Mutations are, of course, the raw material for evolution. That is our trope. Mutation rate estimation is important for things like characterizing the rate of input of novel variation, scaling demographic and evolutionary events, estimating the divergence time of species, and all of those are very important for evolutionary biological reasons, but if you want to talk about things like medical genetics, it also helps us understand the risk and incidence of disease. Now, if you look across the primate clade, as far as we know, mutation rates in primates vary by up to fourfold. And the question that the Pfeiffer lab, among others, tries to ask is why. However, confounding the comparison between all of these different mutation rate studies is a number of different factors. Two of them that are most impactful, of course, is the depth at which these studies use. This on the left is a histogram looking at 35 different mutation rate studies. And as you can see, the average depth or average coverage across the genome varies quite a bit. The second highest one is, of course, the types of filtering that is used to try and find true de novo mutations, or DNMs, that are present in the offspring but not in the parents. There's a wide variety. Now, both of these things can cause big differences in mutation rate estimation, and as a proof of concept, there was actually a publication done in 2021, a study done, where 5 different research groups looked at the same data set and tried to estimate mutation rates in their own specific pipeline, and they got 5 different answers. And the highest and lowest resulted in a twofold difference in mutation rate. Even when you look at simulated data, you can see that, in this case, this is from a previous publication that actually is in revision right now, where we simulated trios of equal coverage at different coverages. So we had a trio of 10x all the way to a trio of 100x and multiples of 10. And we knew where the mutations were, of course, because it's simulated data, and we tried to find them again, utilizing different filter thresholds. And what you can see here is that in those black boxes, those are the recommended filters, and that is the best filter, which means that we were able to capture all of the de novo mutations that we put into it at that level, and also restricted the number of false positives, or FPs, as you can see on the right there. So what this shows here is, of course, as you'd expect with increasing data quality, you get better results, but the best recommended filter thresholds are, of course, different. And also with the differences in data quality across these different studies points to 2 things. One, it means that, as expected, the most effective filter thresholds vary based on the data. And secondly, there needs to be a standard way to properly compare mutation rate studies and base filter thresholds on something. So with that, I am going to introduce this tool that I've been working on called DETECT, or de novo mutation extraction through experimental cutoff thresholds. This is a simulation-based workflow that aims to try and point you to what the best filter threshold is for your data set. And it's really very simple how it works. You have let's say in your data set you have a large curated data set of variants that you want to input into as false positives into your data set. You also have de novo mutations that DETECT can actually put in automatically, or perhaps you have candidates that you want to test on your own. And you put them together into a read simulator. And this is a simulator that creates Illumina reads. And fortunately, as a result of the read simulator, it also inputs things like sequencing errors at a rate that is realistic to Illumina sequencing. That picture on the right there is actually from a previous publication of mine that looks at the sequencing error profiles of several different short read simulators. The one we use specifically is called Mason. Out of that publication, it was deemed to be the best and most analogous to Illumina error profiles. Here in the middle, you can see that this would just be an example of what DETECT would generate from this simulation. You have inherited polymorphisms and sequencing errors that act as noise, and then you also have de novo mutations just in the child. Now DETECT tries to find them again. And now we have a pretty standard pipeline here where you have the simulated reads. They're mapped using BWA-MEM, and then you have very much a GATK best practices pipeline, calling variants, and then eventually isolating it down to what we call Mendelian violations, or MVs. These, in this case of looking at humans and non-human primates, you look generally at things that are sort of novel, meaning that both parents are homozygous reference and the child is heterozygous. So those would be what we call our DNM candidates. So once we have these isolated MVs, or DNM candidates, It then goes through a series of different filters to try and figure out what the best one is. These are the ones that have the best recovery of known DNMs, because of course this is simulated data, so we know where they all are, but it's also the most stringent. So in this case, you can see that with different GQ filters in this case, a GQ of 15 is the best because it is the most stringent, and yet it also recovers all known DNMs. So it output this as a recommended filter. Once you have this recommended filter, it shows a bunch of other things, such as the number of DNMs that were input that were actually recovered. This is known as your true positive rate. It also shows you the false positive rate. We know where the sequencing errors are, and we know where the polymorphisms are. So if these things accidentally make it through the pipeline, we can also tell you which ones made it through and how many did. So that's what we call our false positive rate. And on top of that, of course, because we know where the DNMs are, we get what's called a false negative rate, the things that are missing from our data set. Now, of course, detect can be deployed both ad hoc and proactively. If you have a data set and you want to know the best way to estimate mutation rates, you can use the relative parameters that you can make your simulation as analogous to your real data set as your simulated data set. And then you can also say, how much novel data do I need to estimate mutation rate in my species of choice? You can run detect multiple times at different depths, at different Illumina sequencing technologies to get a better idea of what the best data set is for you. Detect utilizes a bunch of different filters. There's very common ones that are used in mutation rate estimation, such as allele balance in the child, basically determining looking at how heterozygous the child is at that site. The genomic depth, low or high coverage, can be indicative of low power or something like a paralogous collapse in the reference genome. Site quality, genotype quality, and of course, having as few reads as possible that point to the alternate allele in the parents, because that can imply a miscall in the parents. On top of that, what we actually showed in our previous study is that in the GATK best practice filters, which is known as the hard filter, in low coverages, it can actually throw out true de novo mutations. And so what we actually did was broke this down into its constituent parts because it's multiple filters put together, and so each one of those can be tested independently as well. When you look at something like a user experience, there's a lot of moving parts because we want to make this as analogous to your data as possible, or so that you can make it as analogous to your data as possible. But you give it things like, for example, polymorphisms that you want to embed as false positives. You give it a reference genome, you give it the read length, fragment length, et cetera, of your Illumina technology that you're working with. And the final thing that you do is you actually input, among other things, an a priori mutation rate, or you can give it a VCF of mutations that you want to embed as true positives as well. And then you have what's called a filter file. This is just something that I've calling it. For example, this bottom left would be an example of a filter file where you have a minimum, a maximum, and a step size with which the filter you want is going to start iterating through. So in this case, with GQ and that green box in the bottom left, it's going to range from 5 to 20 in steps of 5 and try and filter. And it will return one of those values as considered to be the best. That is the one that filters all of the DNMs, or that keeps all the DNMs. Once that's output as a JSON file, it then gets put into the detect workflow, which is implemented in SnakeMake. And then you get a list of VCF files that are then aggregated across iterations, if you so want to run iterations. And then it generates this sort of output here that you see in the bottom right, with the average recommended filter from each of the iterations, the variance, and the value at each one of the iterations that was recommended. So how effective is it really? In order to test how effective it was, we used a data set with real validated novo mutations via Sanger sequencing. This is from Venn et al. 2014. We were looking at it. This is looking at multiple chimpanzee trios, and we looked at 2 individual trios in here. Just called them Trio 1 and Trio 2. Across the board, they had 60 validated DNMs due to reasons based on reference genome accessibility. We actually left these over, so we lost one of them. However, we then input these sort of values, these coverages, these read length, et cetera, into detect, and then tried to see how good it was at recommending the best threshold filters. Now, of course, here are the results. Each one of these was a recommended filter threshold and then applied in sequence. So on the left here, we have what are called the best filters. These are the ones because we know which the DNMs are. We also know which values would be best at filtering them. So on the left here, we have that if we were to have perfect knowledge of all of the filters, and then on the right, we have the detect recommendations being pulled through. Now, as you can see, while it's not perfect, it gets the number down from 34,000 to less than 150 candidate sites, which could then be moved on to manual curation, and we end up losing 3 true de novo mutations. On the right, trio 2, on the right you see the same thing happening where it is not as good as the best filters. It is not as good if we had knowledge of the ground truth. However, it gets it down from 34,000 to under 100. In both of these cases, the idea of detect, of course, is not to get it down to a perfect data set, but rather to get it to a data set in which you can then manually curate and make sure, and you can get a better idea of what the true de novo mutations are, usually from a visual genome finder like IGV. And in addition, this trio actually has what we call a higher DNM proportion. That is to say, at the bottom, at the end, you can see that detect recommendations have 21 out of 84 of the final sites were true de novo mutations, which is 25% of the data set, which is actually higher than if we were to use the best filters. If we were comparing detect to other things that are on the market right now, Rates Tools is an example of this. It does something a little different where it outputs a mutation rate estimation. However, it has high numbers of dependencies, and utilizing the same exact data set as Venn et al. 2014, they actually only recovered 63% of the DNMs, with 11% of the DNMs making up the final candidate list. However, in Detect, we only have five required bioinformatics softwares. We recover 92% of the DNMs, and we have almost double the DNM proportion in the final candidate site. So not only is this effective at isolating it, it's also competitive with other tools that are doing similar things. So right now, it is not out yet, but I'm in the process of doing benchmark tests as we run this. We have a manuscript in production right now, and we have many different planned updates, such as updating it to SnakeMake 8, including support for long-read simulators, reducing the number of dependencies, of course, increasing sensitivity, and improving user-friendliness. So with that, I would like to thank the Pfeiffer Lab, of course, Dr. Suzanne Pfeiffer, my mentor, Dr. Eldad Urivias, Cheryl Versosa, Suhail Gafour, our IT guy, who really helped me with SnakeMake because it's not an intuitive program, and McKenna Sai, of course, and also my committee members, Dr. Jeffrey Jensen, Dr. John McCutcheon, and Dr. Fritz Sedlacek. Suzanne also wanted me to let you know that she's hiring postdocs for this lab. If you're interested in mutation rate and recombination rate variation in non-human primates, we've got a lot of cool data coming up. So talk to me and I can get you in contact with Suzanne. Speaking of interesting new novel primate stuff, we have 3 other talks that are going on right now looking at II genomics, looking specifically at crossover non-recombination rates with Eldad. He is going to be in that room right there. Dr. Turbot, inverting demographic history, and Cyril Versosa looking at structural variant. And thank you. I'll be taking questions.
All right. Good afternoon, everyone. My name is Andrius Dagilis. I'm an assistant professor at the University of Connecticut, the Department of Ecology and Evolutionary Biology. I just started there, but today I'll be telling you about a project that was kind of a swan song as I was leaving the Daniel Matute lab. Thank you all for joining me in your post-lunch stupor. Hopefully I will make this fun and exciting for you so you stay awake. So first of all, what is coevolution? I'm actually going to be talking about gene-gene coevolution, so if you showed up here hoping I'll tell you about relationships between species, I know nothing about them. Gene-gene coevolution, at least for purposes of this talk, I will define as the covariation between the rates of mutation in 2 different genes. So here, for instance, we have 3 different genes. Gene 1 and gene 2 have mutations indicated by a star somewhere in the phylogeny, and they seem to be covariant of where they're occurring, whereas gene 3 has mutations entirely in different branches of the phylogeny. Early developments for these methods were made by Nathan Clark and colleagues. They've been used in a variety of systems, but the general idea is what you do is you build a bunch of gene trees. You transform branches in those gene trees by dividing their lengths in the total species tree. This helps account for stuff like demographic changes or changes in genome-wide mutation rate on different branches or evolutionary rate in general. And what you get out is a rate of evolution on each branch for each gene, and you just correlate them. So in this case, gene 1 and gene 2 have high correlations between each other, so does gene 1 and gene 3, but gene 2 and gene 3 don't. With 3 genes, this is pretty simple. As you increase the number of genes you're including in the data set, very quickly you end up with a normal combinatorial nightmare of, for instance, a study in yeast that looked at about 2,000 genes, had to look about 3 million combinations of those genes, right? But what they find is, hey, there's a lot of coevolution among these genes. So we thought, hey, let's look at this in Drosophila, where we have a bunch of species, we know what they're, we have a bunch of orthologous sequences. Ideally, right, you build these gene trees in a way where you're not using paralogs, you're not using stuff that has multiple copies. So in our case, we had a bunch of Buscos. Let's go through them and see what you can do with it. Can we build a nice little pretty network like here, this is the yeast network. Can we build something like that and build a nice network for Drosophila? So first of all, we were just going to ask how much gene-gene coevolution is there in Drosophila. But part of the reason we wanted to do this in Drosophila rather than another system is we wanted to know how informative are these correlations that are happening across really large macro scales into what's happening in individual species, right? So Drosophila melanogaster as a model system, we have a lot of information about what's happening within species. We have candidates for speciation genes, for introgression pairs, all sorts of other things, and we can ask, does this network actually correlate with what's happening within species rather than just looking across the broad thing? And finally, something that I've always kept in my mind throughout my career is I love structural variation. I love thinking about how the genome is organized. And when you have these things that apparently are going to be co-evolving across huge swaths of species over long evolutionary periods of time, those seem like prime candidates to look for stuff where, hey, maybe they're causing genome to be organized. So first big result is that Drosophila genes co-evolve a lot less than fungi. So I showed you this plot before from Stenwick et al. 2022. Their average gene-gene correlation is 0.57. Ours is 0.055, right? This is among 2,800 Buscos in 155 species of Drosophila. Far, far less coevolution. You might say that's a problem, but actually for us that was kind of a win. They had to really choose a very stringent cutoff to kind of narrow down of the interactions of interest. We just took the significant ones. Makes life easier. So in total, there's 70,000 significant interactions among these Buscos. That's roughly 2% of the 4 million interactions that we were looking at. There's significant overlap with these and known physical and genetic interactions, so that's kind of a nice quality test of like, okay, we're capturing something real. But that's kind of where the sense started breaking down. So in this network here, what I'm showing, each point is an individual Busco, right? Edges between them are the strength of correlation between them. And I've colored them by communities, which we'll talk about in a second. But the first thing I'll point out is all of these genes across the edge that just don't co-evolve with anything. Some of these are going to be probably because they're relatively low copy, well, probably because they're found in relatively few of our 155 species, so we have lower power to detect coevolution between them. But for some of them, they're present in 153 of the species and just don't seem to co-evolve with anything else. Right, so that's already a little bit unusual. Second, if we do look at these 4 communities, those 4 communities, you know, all of us the biologists, we see things clustering together. We kind of go like, oh, there has to be some meaningful thing that's linking them together. I've tried to skin this cat every single way imaginable. They do differ in terms of their single-cell RNA expression profiles. On average, clusters 3 and 4 are expressed slightly more highly across a wide array of tissues than either 1 or 2. That kind of might make some sense because cluster 1, you can see, is this giant set of a bunch of genes that are all co-evolving very weakly with each other. Cluster 4 is a small cluster of genes that are all very tightly linked with each other. Okay, so highly expressed things maybe are more likely to co-evolve with each other than not. But the effect sizes are pretty small. And more importantly, if we do something like a Go analysis, nothing, right? There's absolutely no enrichment here. Part of the problem is we're looking at a small subset of genes in Drosophila in general, right? So it's 2,800 Buscos. This first cluster is 1,800 genes. It's pretty hard to get Go enrichment when you're taking half your data set and asking, okay, what's enriched here? Probably not much. But surprisingly, we don't find it even in these smaller clusters. If we relax Go assumptions and do the wrong thing, and so I'm telling you this is not Go enrichment, but it's just giving us an idea of what these clusters are, and we ask, okay, among all Drosophila genes, what do we find that's related here? Well, we do find kind of meaningful, some kind of descriptions that seem to align with each cluster. Out of the top 10 Go categories for cluster 1, 8 of them are related to metabolism. Out of the top 10 for 2, 6 of them are related to biosynthesis. Out of the top 10 and 3, you get organ development and regulation, and 4 still only has one thing, which is it's found in cytosol or cytoplasm. So these clusters, clearly they relate to something because they're both related to physical interactions and genetic interactions. Clearly there's differences in expression, but there's not a super clear biological link here with here's what they are. Here's the genes that are co-evolving. Everything that's in a particular location is doing it. So that was a little bit frustrating because, you know, instead of just being able to replicate someone else's pipeline basically and being able to say like, hey, it's cool, Drosophila has a bunch of coevolution, and here's the exact things that co-evolve. It has coevolution, but it's not as easy to interpret. But on the other hand, we still had all this data within Drosophila melanogaster where we could ask, okay, do the things that co-evolve actually lead to meaningful differences in what's happening within species? So in particular, I immediately thought, when I thought about doing this analysis, about John Poole's data set of looking at potential incompatibilities within Drosophila melanogaster that are segregating throughout the population. Hey, you have an interaction network where you have a bunch of places throughout the genome that you have potential incompatibilities. How strongly do these overlap with the interactions that we find? They don't. Part of a problem is relatively few buscos fall into those regions that they identify, right? We're not looking at the entire genome, we're looking at a smaller subset of genes. So then I went back to the drawing board and I thought, okay, do we have data where we can just look at exact busco locations and see what's there? Well, Jen Coughlin and I had just recently done a giant analysis of melanogaster across the world. And we had identified a whole bunch of potential genomic ancestries within these. We had identified introgression among these ancestries. So I thought, what if I constructed a new kind of network? And I think this is why this ended up in the methods section. This is the softest method I can think of. Hey, let's think about genes that co-introgress. So each of these points is an arrangement of free populations, free ancestries in Drosophila. And we measure introgression in terms of D statistics. In this case, we used a DF statistic, but pick your poison. And we can ask, okay, how well do these statistics correlate between two different genes? If two genes are highly correlated in their introgression statistics, that means that whenever they do introgress, they both introgress at the same time. If they're negatively correlated, it means only one of them might introgress. And that by itself is, I think, an interesting measure that we tend to not think about in the introgression world. We usually try to think about, like, what are the things that are introgressing as individuals? But introgression probably doesn't happen on a unit scale of just a gene. Often it'll happen as a whole inversion or a whole chromosome arm or maybe an entire chromosome. And so trying to think about the different genes that come together as they introgress is, I think, something that we all should be doing more and my lab is expanding more into. Anyways, out of this, doing this again, we find a roughly similar number of significant interactions. About 60,000 pairs of genes are significantly co-introgressing. And I know it's really hard to see, but over there's a little bit of negative co-introgression. Those are interesting genes, right? Those are gene pairs that either one or the other introgresses, but not both. On the other hand, when we compare it to coevolution, we again kind of hit a dud. So on the x-axis here is this branch length correlation that's measuring how correlated evolutionary rates are across the entire Drosophila set. On the y-axis is how well they're correlated in terms of their introgression. Okay, this is a statistically significant link, and I've done the simplest test here, but I've, again, done this many different ways, and yes, sure, there's an influence, there's some kind of statistical link between these two things, but the effect sizes, no matter what you do, are tiny, right? And indeed, what you find is that the genes that most strongly co-introgress very rarely are ones that are strongly co-evolving across all of Drosophila. So this is kind of like I feel like the theme I've seen in this conference has been, we all talk about, like, can we predict evolution? And the answer is yes, but barely. So yes, we can predict which genes are going to co-introgress, but just a tiny, tiny bit based off of what's happening in terms of coevolution. The last thing that was interesting to me about this data set is we can go and ask, okay, where are these coevolutionary interactions taking place? Because in a lot of ways, we might expect things that are co-evolving over very long periods of time to be brought together by structural variants or all sorts of other things. And so here I have a map of Drosophila melanogaster, where each of these points on the outside is a single gene in this busco data set. They're colored by their communities. Their heights are relative to their degree, so how many significant interactions they have. And it's a mess, right? There's not a super clear pattern here. Importantly, there's no enrichment of any of these communities within each of the chromosomes, right? You don't see a pattern where if you're in some community, you're also maybe located somewhere. That was the other thing that I thought of, like, oh man, we have 4 communities and there's 5 molar elements, so maybe if we just drop one, they all just cluster to different molar element. Nope. And indeed, what you find is there is a slight increase of the significance of an interaction if you are on the same chromosome. One way of portraying that here if you're looking at pairwise chromosome pairs, the size of the dots here is the proportion of them that are significant, and the color is based on the average correlation. You do see a pattern where, for instance, pairs of genes on the X tend to co-evolve much more strongly than pairs of genes among other chromosomes, but it's not a terribly, again, significant pattern. The difference is roughly 2.1% of interactions on the same chromosome are significant versus 1.7 between. We find the same for co-introgression, 2.4 versus 1.6, right? Both of these patterns are somewhat driven by location, but not entirely. And if we further look within each chromosome and ask how far apart you are on a chromosome, how much does that determine whether you either co-evolve or co-introgress? It is significantly correlated with co-introgression. Yay, that makes sense. Things near each other in chromosomes should co-introgress. It's not with this gene-gene branch coevolution. Of course, we have more information just melanogaster, right? I'm looking at 155 species, but sadly only 13 of them we actually have data for in terms of chromosome scale assemblies. So in this case, I'm just showing how that pattern is mirrored across all 13 species. You do see same chromosome will tend to show more significant interactions. Indeed, if we plot it across the phylogeny, you find a weak relationship where it seems like most of them it's pretty much the same except for 2 outliers. And the interesting thing about those outliers is they show the opposite trend in terms of their slopes. They show the, so Seychellia, which shows the weakest effect of being on the same chromosome or on different chromosomes, shows the strongest effect of how far apart on chromosomes you are, whether you are co-introgress, or whether you're co-evolving or not. All right, so I'll very quickly wrap up. Whether 2 genes co-evolve or not is not a strong indicator of any particular process within species. It's weakly associated protein interactions, that's also true in yeast. Genetic interactions, also true in yeast. I didn't talk about LD, but it's also associated with LD in melanogaster, with co-introgression in melanogaster, but also all of these could probably just be explained by the fact that they're close to each other. Can we do better? Probably, by including more genes. By looking at different networks, right? Drosophila just seems to show very little coevolution. Maybe other organisms don't. So if you're interested in any of these questions, please join the lab. I'm looking for postdocs and graduate students. Find me at the conference or email me. And I barely have time for questions, but if you have a question, go for it.
We see how the slides go then. So hello everyone. Today we'll talk about methods that are commonly used, mainly in landscape genetics, to infer the landscape drivers of gene flow. And with my collaborators, Marie-Josée Fortin and Pedro Perez Neto, we wanted to compare 2 methods, one that is commonly used and one that we developed for the sake of this study that is based on spatial eigenvectors, aka Moran eigenvectors map. So I will also take the opportunity with this talk to talk about the advantages and pitfalls of this kind of inferences because I've been working on them during my PhD and I keep studying them and I want to stress some of these points. So let me first recall the fact that habitat connectivity brings forth the dispersal process, and this often contributes positively to long-term population survival. And this is why habitat connectivity is central to biodiversity conservation. But if we want to implement efficient conservation measure for connectivity, then we need to model it. And modelers, when they do that, they often rely on maps of habitat patches and on parameters about how species move from one patch to another. And the common way to do that is to assign a cost to different types of land cover, assuming the cost that is associated with a movement through these land covers. And once we have done that, we can compute what we call the least cost path. So in that example, this is the path connecting the green spot and the red spot, minimizing the total sum of the cost over the pixels. So this is what we call the least cost path, and the associated sum of the cost is called a cost distance. So it's a distance weighted by the cost of movement. So here, what will be critical for the reliability of the output of such a modeling is the way in which we assign the cost. And for that, we can use landscape genetic approaches. So we rely on the fact that neutral genetic patterns depend on genetic drift and on gene flow depend on dispersal, hence on the parameters I mentioned before. So there is a way to use genetic distances to look at their correlation with cost distances until we identify the best scenario, the scenarios that will maximize the fit of this relationship, and then we'll assume that this is the one that best reflects dispersal. So for doing that, we commonly use distance-based models. So they fit a statistical relationship between two matrices, a genetic distance matrix and a cost distance matrix. And for that, we use linear mixed models, having a population-level random effect to account for the lack of independence inherent to pairwise data. And we do that for several pairs of distance matrices because we will test different costs scenarios, different candidate cost scenarios, until we identify the cost scenario bringing the best statistical fit. And we'll assume it's the one reflecting best reality. But this method has several limitations, and I will mention two. First of all, when we have complete distance matrix, they can't capture the complexity of dispersal patterns emerging from especially heterogeneous population networks. In this example, we see that when there is a certain degree of clustering in population, after a given distance, the relationship between cost distance and genetic distance plateaus. And when we have this kind of patterns, we need a multi-scale decomposition of the signal if we want to make sense of what is happening and identify the true driver of dispersal. The second limitation we have with cost distances is that if you consider a reference cost distance matrix and you look at how it's correlated with other cost distance matrices computed with totally different cost scenarios, they often exhibit a super high correlation. So on the plot here, I use this cost distance matrix as a reference and in green. I computed the correlation with other cost distance matrices computed with different cost scenario, and I could see across a lot of realistic landscapes that the correlation was super high. So many cost scenarios, totally different qualitatively, bring high correlation. So in that case, it means that we also need to disentangle what is the cost component from what is the distance component in this cost distance matrix. Otherwise, we can't make sense of the signal. So one potential solution to this is to use multi-scale decomposition of the distance matrix. So for that, we decompose these distance matrices into spatial eigenvectors, and that's what we call Moran's eigenvector maps. The advantage of this is that the vectors we obtain, they are defined at the population level, and so we have several vectors that are zero correlated, so they are totally orthonormal and independent. And on top of that, we have them at the population level, which prevents the issues we have with distance-based models, so pairwise data. So with this kind of data, with this format of data, then we can use redundancy analysis, a method that is commonly used in community ecology to relate population level or community level responses to environmental parameters. And we just need to also convert the genetic signal into a set of eigenvectors. And then we can relate these two signals to see what is the scenario bringing the best fit. So although this approach seems promising, it had never been tested before, so that's what we wanted to do, and we wanted to compare these two kinds of models in their ability to recover what is the true scenario driving dispersal. So we compared their relative fit with different cost scenarios to identify which one was the best. The issue here is that if we don't know the truth before, we can't really assess them, so for that reason we use the genetic simulations. We randomly sample 50 landscape within a land cover database in North America across forest cover gradient. And within each of these landscape, we randomly sample 50 populations in forest patches. We had 6 land cover types, and we assigned them a cost value, which is here the reference cost value. So this is the truth that the model were supposed to recover afterwards. And with that, we computed the cost distance matrix among these populations. We also used this cost distance matrix to simulate neutral genetic patterns that were driven by genetic drift, which was constant everywhere because each population, its individual population had 30 individuals. And then gene flow depended on the cost distance between population as given by the cost distance matrix. And we obtained a genetic distance matrix in each of these landscapes in the end, which was the signal we could use to recover the truth. But in parallel, to really assess finally how these methods perform to recover that, we computed 100 other cost distance matrices using random cost scenarios. So we reshuffled the cost values across land cover types to generate artifactual scenario and other cost distance matrices. And we assessed to which point each candidate's other cost distance matrix related to the truth. So we had the distance from the truth of each other cost scenario. And finally, we modeled the genetic distances using these two other approaches in order to identify which one was the best. And the rationale of what we implemented was to assume that a good model will assign will have a good goodness of fit when using a cost scenario that is close to the truth. So because the goodness of fit values in themselves were not comparable, we compared the rank of similarities of cost scenario to the rank in terms of goodness of fit of each scenario for the two models. So we, in the end, had a Spearman rank correlation that was a measure of inference quality, because if it was high, it meant that the model had a goodness of fit for scenarios very close to the truth. In the end, what we saw, unfortunately, is that distance-based models were still the best in almost all the landscape. In 78% of the cases, they were the best. So there were still landscapes where spatial eigenvectors provided a better inferences. And for finer assessments, we looked at the scenario providing the 10 best goodness of fit values. And the good news here is that these cost scenarios overall gave cost-distance matrices highly correlated to the truth. In average, it was above 0.8. However, in almost all the cases, there was one cost scenario giving a cost-distance matrix not correlated at all to the truth. That was also among the 10 best scenario. And the true scenario, when we compared the goodness of fit for the true values, it was among the best models in only half to 2/3 of the cases. So in summary, the method we studied did not really improve the inferences, except that in some landscapes it could still provide a better method. So this method deserves further investigation, and we should identify in which landscapes this method performs better, and we should also better understand which kind of scenario we can distinguish from each other. And in that sense, the method we developed offers some new perspective, because we could use the advantage of redundancy analysis to decompose the cost distance signal into a cost component and a distance component. And this might not improve how we perform the inference. However, it could improve our understanding of the power we really have when we do this kind of inferences, and we could understand why it often doesn't work. So I just want to finish with the fact that what this study shows and means is in accordance to what we have showed in the past, meaning that this kind of inferences should be interpreted with a lot of caution. And in many cases, it's better to look at multiple burst scenarios instead of looking at the one scenario providing the best goodness of fit. And on top of this, we always have to remember that these scenarios are often translated into corridors, and the value we infer have a lot of consequences for the location of the corridors. So we should be careful with that. With that, I would like to thank my collaborators for this study, and to thank you all for your attention.
Good evening, my name is Pany Decossard, and the topic I'm going to present today is on the geological origin of biochemistry. And this topic is, the theme is part of a book that I published last year on Ways of Nature, how the trail of life exposed the universe. And so what I'm going to talk is basically first some theoretical considerations more than empirical considerations based on studies that have already been published. Basically, we'll talk about that life did not start with cells, and life did not start with self-replicating RNA molecules. And I'm going to suggest the hypothesis that life started with autocatalytic systems of RNA and proteins, and in the environmental context of minerals. And if we look at displayed here, the core elements of life. If you look at any systems like cells, those are the core elements of cells. Basically, nucleic acids and protein. Protein here represented by a polymerase that replicate or transcribe nucleic acids, and the relationship between those different elements, and none of those elements happen spontaneously. They are all catalyzed, and the last element is always catalyzed by a different one. So nucleic acid, the synthesis of protein will be catalyzed by RNA from the ribosomes, and the processes of RNA and DNA are going to be catalyzed by protein, and that make a circuit. And however, when we look at the theories about the origin of life, it suggests that life started as a self-replicating RNA molecules. RNA would have been brought about by prebiotic synthesis and self-replicating and starting the first genome of life, kickstarting life. However, when we look at all life systems, there are no life systems that where nucleic acids are replicated by nucleic acids. In all life, from viroids to man, nucleic acids are always replicated or transcribed by protein catalyst, and protein are always synthesized by RNA catalyst. And here we have the COVID virus RNA in red. To make an offspring in yellow, so we need the non-structural protein 12, NSP12, to catalyze that transformation from RNA genome, from a COVID parent to a COVID offspring. And the same thing for proteins in the ribosome, which is the translation from the messenger RNA is always catalyzed by the ribosomal RNA. And we know the ribosome is constituted by protein and ribosome and RNA, but it's the RNA component, as we know, that is responsible for the catalysis. So in other words, life started as an autocatalytic nucleoprotein system, not just as RNA. And that brings us to our definition of life that we had in our book. Basically here, mostly talking about autocatalytic system based on proteins and nucleic acids, so not just RNA, according to our proposition. But based on that definition of autocatalytic ribonucleoprotein systems, how can we identify the origin of life from that definition? And so we start here by the theory that was proposed a while ago by the physician biologist Strothofen, who suggested that life started as autocatalytic systems without any further specification, given a food set, and we add plus environment, given a food set autocatalytic system. And of RNA and protein that, given a food set of amino acids and nucleotides, would continue to perpetuate and evolve. And now we go back to the emergence of life, would be nucleic acids, genomic RNA, and a ribosome, and a protein, RNA polymerase, and the relationship between genomic RNA that would translate into a protein, RNA polymerase, and under the catalysis of ribosome, and that same protein also involve RNA polymerase also get involved in the synthesis in the processes of RNA. So it's basically that life is basically a circuit based on those two elements, basic elements. That's the simplest component of life without a cell. This is the circuit. And some of the conditions that needed for the emergence of life, we have in the diluting environment of life, in the aqueous environment of life, we need a system for compartmentalization, encapsulation, a protecting environment. We need polymerization, homochirality, and information and genetic code. So life started, as we said, as autocatalytic nucleoprotein system, but where is that? Now, all point to mineral surfaces, and we come up with that with the system. Well, the black box is mineral surfaces, and nucleotides and amino acid would be the input. And that would lead to RNA and ribonucleoprotein system that constitutes the emergence of life. And mineral surfaces, basically the mostly studied in life emergence is clay minerals and LDH. Mormogellonite particularly studied quite well. Here, mormogellonite in nature. And the basic structure of clay minerals are basically tetrahedral units. From here, on the corners, we have oxygen for the tetrahedron. In the center, we have silicon ion, quadrivalent silicon ion. And for the octahedral sheets, we have aluminum in the center, aluminum cation, or sometimes could be magnesium. And in the corners, you have oxygen, or can be also hydroxyl groups. And here, the basic structure of mormogellonite or iliate. In the center, number 2, you have an octahedral sheet, which is sandwiched between two tetrahedral sheets up in the bottom. In the tetrahedral sheet at the bottom, the dark dots are silicon ion, and then in the octahedral sheet in the center could be the aluminum or magnesium. So the minerals can swell, can absorb water. Here on your right side, you'll see the mineral here, the different sheets, and then you have the layers. And in between, you have in the interlayer regions, you have a lot of water. can absorb a lot of water, and you have a lot of cations, sodium and calcium, so they can swell. During the wet and dry cycles that they can have in hot springs, they can swell, and it is in the interstices, in the interlayer regions of the minerals that we suggest that those autocatalytic nucleoprotein systems were formed. Here, those are we'll review those elements one by the other. Those are the box that mineral surfaces need to check to be the cradle of emergence of life. So it has to be a protecting environment, polymerization, homochirality, information, and genetic codes. So mineral surfaces check that box of concentration. Mineral surfaces, clay minerals, have been shown to adsorb, concentrate, and protect the monomers of life, amino acid or nucleotide, particularly protect them against UV radiation. And the LDH, which is a mineral that was common in early Earth, according to a silico study, shows that this mineral can swell, adsorb, also concentrate basically amino acids. Clay mineral for both nucleotides and amino acids, LDH only for amino acids. And clay mineral also can polymerize. Both of them can polymerize and form the bond that are necessary. For forming RNA, you need the phosphodiester bonds, and the peptide bond for the formation of polypeptides. So that studies appeared in 1996 as a letter to Nature from James Ferris and collaborators, where it showed that here we will describe a system that models prebiotic polymerization by the oligomerization of activated monomer, both nucleotides and amino acids. So early on, he showed that those minerals, he used mormorillonite and iliad to show that they could polymerize nucleotides and amino acids. And he showed that with different successive feedings, it could go up to 40 mers of RNA or nucleotides or amino acids. And this is the silico study that showed that mineral surfaces also can promote the polymerization of amino acids. And that polymerization of amino acids in LDH is done as it happened also in the ribosome from the N-terminal to the C-terminal. So there is information that passes from the minerals to the synthesis of proteins in the ribosome. And homochirality also as well. It has been shown that mineral surfaces, they can preferentially absorb one stereoisomer over another, whether the D or L isomer. And we know for like in life, the proteins are made mostly of L amino acids, and the ribonucleotides mostly of D sugars. And it has been shown that mormorillonite can preferentially absorb one of the isomers. And when in the studies of Ferris, if he has a mixed D and L nucleotides, and the mineral surface will make dimers which are DD or LL instead of having DL spots. So they will combine the same stereoisomers together. Information, how does CAC provide information? When also he tried to make dimers of the amino acid of nucleotides, if you put equimolecular amounts of the 4 bases of RNA, A, C, G, and U, so that would give you about 16 dimers, 4 to the power of 2. But 8 dimers would form about 84% of the 16 possible dimers, and that's information, that's non-random. And also 8 of those dimers would start, all of those dimers would start with a purine nucleotide, either adenine or guanine. And in mRNA, in real life, the messenger RNA always starts with a bunch of purine nucleotides. Information as well, as I said before, in the synthesis of amino acid or proteins by LDH, always proceeds from the N-terminus to the C-terminus, as is also happened in the ribosome. But in the mineral surface, as we said here, we have silicon cation, which is basically completely homogeneous, or we have aluminum or magnesium ion cation in the octahedral sheet. How does information come from that? Because there's no specification, it's completely periodical. But there is that element, so it's the imperfection of the crystals, mostly what they call isomorphous substitution, where in the tetrahedral sheet at the bottom, the cation can be replaced by lower valent cation. Now, the silicon can be replaced by aluminum. Silicon is 4+, can be replaced by aluminum, by ferric ion, or by ferrous ion. Or in the center, it can be replaced by the hydroxyl or magnesium can be replaced by lower valent cation. So we think this is that isomorphic substitution would be responsible for biological information. And is this for the genetic code as well? And we think also that if a certain segment of the mineral can synthesize can add an amino acid to a chain, it could also add a certain codon to a chain of RNA. And that by mathematical transitivity, that relationship will form the genetic code. It's the same thing that we see in the synthesis of protein in the ribosome, where the anticodon is related to a codon, and also related to amino acids. So the mineral surfaces was the anticodon, was the prebiotic anticodon. It can be related to both an amino acid and a codon, and by this mathematical transitivity, establish the genetic code. In conclusion, we'll say that mineral surfaces check all the boxes, fulfill all the requirements needed to be the state of emergence of life. In other words, geochemistry formed the scaffold upon which biochemistry was constructed. Thank you.